stories.
I always have good stories, and I just clicked start. So maybe you
crap.
want to hear some of them. I love to do this tomorrow, just while he's talking about something. Just start the video, so because we don't edit.
Okay.
All right.
Hot in the news today. Oh, wait a minute. Oh, this is Bob, and I'm Mark. How's
The
that?
Grumpy Scientist.
The Grumpy Scientist. And
If you don't like us, move along.
Quit typing in Grumpy Science.
Quit sending
on
us
Google.
nasty grams. Go watch the Grumpy Cats or something. They're a lot funnier than we are.
Actually, we're starting to beat the Grumpy Cat on the Google search.
Yeah,
that's pretty
we're beating
impressive.
the Grumpy Cat pretty well. Well,
Yeah.
I don't think the Grumpy Cat has posted nearly as frequently as we have.
No, but their one post got 10,000 views, and we've just got 4 viewers. Keep watching.
I think it's the
same
same
ones.
2 viewers, and you guys are watching twice, so keep going.
Awesome.
All right,
We're
stick
using
with
fake
us.
names,
That's
so
right.
that works good too.
That's right. Okay, so today, my esteemed colleague, Dr. Tommerdahl, wants to talk about a topic that's a brand new topic. No one's ever thought of this before, and it's all in the news now. It's a big
It's the
deal,
news,
big
yeah.
splash.
Actually,
right?
it got me grumpy driving into work. I was listening to the news, and I was like, Johnson & Johnson has announced here in their phase 3 clinical trials of a form of ketamine for treating neurological disorders such as schizophrenia.
And depression.
And
things like
depression,
that. Yeah, chronic pain,
OCD,
maybe.
and a couple of other things. Well, first let's tell people what ketamine is. And ketamine is an NMDA receptor antagonist.
Broadly speaking, though, ketamine is
an anesthetic. It's used
right?
very often, and
it's
if you
used a lot in
surgery,
veterinary too,
it's used in
right?
people.
In
I mean,
people,
you break your
it's
arm,
animals
and
and people. It's
just
just general anesthetic, right?
Yeah.
It just puts you out pretty much completely.
or it does very. You can take all levels of it. You don't have to get put out completely.
Right, but I mean, it can. Right.
It's been used for a lot of things.
It's not a local.
No,
right?
it
acts centrally.
It's acts
because
centrally.
it is an NMDA,
NMDA
receptor
antagonist. NMDA stands for you nerds out there,
That's right.
N-methyl D-aspartate.
Right.
And-
Rated S for science. It's getting science.
Okay, so we give you a little bit of geeky stuff here. Okay, so basically we have talked before. For you, those of you who have skipped all the other videos, go back and watch them.
That's
But
right.
we've talked
Do your
before
homework.
about how important it is to have a balance between excitation and inhibition. And we've talked a lot about GABA and GABA agonists and things like that. Well, GABA is sort of on the inhibitory side.
Mhm.
NMDA receptors are on the excitatory side. They're usually excited by glutamate. They are the most predominant receptor in the upper cortical layers. So this is something we studied for many years in the lab. And basically, it's what's called a voltage-sensitive or voltage-dependent receptor, so that you have to have, like first time it feels something, it's kind of like, I'm not impressed. Second time, it's like, well, this is getting interesting. So you keep repeating a stimulus, and it gets better and better and better.
Yeah.
And for those of you who have a brain gauge, these stimuli last a little while, half a second, but it's multiple stimuli, and that's the reason. That's one of the reasons. Well, we have a lot of reasons for doing it that way. But anyway, as the NMDA receptor antagonist gets a little more activated, let's say you get 2 stimuli, one beside the other. Oh, this way I'll point to them instead of you.
That's right. I already know this.
Okay, so we got 2 stimuli. Let's say you stimulate 2 places in the brain. You got one that gets a little more activated than the other. And then the next stimulus, it makes it a little more active. But now that it's more active, it is driving this one down.
Lateral inhibition.
Now, the one on the left side, those NMDA receptors no longer are active because, well, they don't care. Their activity levels are lower. So the next stimulus, all of a sudden, boom, it gets bigger. And so those things are really good at differentiating things. And we've also talked about one of the things that we measure and talk about the most are the building blocks of information processing. We don't test these big global specific things like schizophrenia or OCD or whatever, but we have done studies with all these different neurological disorders. And the reason our measures are effective is because when the building blocks are shot, you have error propagation, and all of a sudden that error propagation leads to something going wrong. And whether that something going wrong is, autism or schizophrenia or some other developmental disorder or degenerative disorder.
Some behavioral thing you can
behavior.
measure by observing someone and checking off boxes to, according to the diagnostic and statistical manual.
Yeah.
They have certain behaviors, that person, and they kind of have to observe you under certain circumstances and stuff. And that's not really the kinds of stuff that where Mark studies directly. He's looking more at like what are the mechanisms going on in the cortex and how are these things interacting in the brain that may impinge upon, may contribute to in some way or another, abnormal function.
Right. And you sort of think about it in terms of they're functional units that are side by side, and if they're all disorganized in a different way, then you get something that looks totally colossally different.
Right.
And when I say building blocks of information processing, just think about how would a structure change if you just changed the shape of the concrete blocks that are used to build it.
Exactly. So you can think of the individual pieces, right? And if you've got all these odd-shaped concrete blocks and they don't really fit together that well, right, then you got to kind of do things to compensate for that. But then you can also think of it as a network, right, which is another thing that the brain gauge in Mark's research is kind of probing, which is not just how these individual structures look and how they act, but how they interact with other things at higher and higher levels, a sort of fractal organization Mark's talked about a bunch of times. And so that's the sort of thing that Mark's research is looking at. Right.
And so the information we heard on the news today was, and I guess we can say NPR because I don't know, everybody, it's public.
Yeah,
and so they were reporting that Johnson & Johnson had released this public awareness blog or whatever.
A PR campaign to get you to line up.
Because they're in their
talk to your physician about.
They didn't invent ketamine, but they made a nasal form of it that's in the 3rd phase of clinical trials.
Right.
And it's not approved for all these neurological disorders, but it's approved as an anesthetic. And so people are using it for other things. People are prescribing it for other things because people that have all kinds of different disorders, they might have ADHD for a while and it morphs into something else and they can't think clearly or whatever. Well, basically, as I tell my students, it's all about balance of excitation and inhibition. And 50% of the correct answers on their test is GABA. The other half has to do
right. If in doubt, check off the GABA box.
Check off the NMDA receptor box on the other side. And basically, it is an interesting thing to listen to. So, you don't have to listen to us. You can go listen to somebody knowledgeable talk about it on radio.
Somebody knows what they're talking about.
Go listen to their podcast. But the reason it got me grumpy was because I was thinking, wow, really, you guys now are acting like this is new?
Well, this is really, an average person couldn't possibly know this. But when you're a scientist and you really know what's in the news. By the way, I've got an intense grumpiness thing I'd like to talk about that's exactly like this.
Yeah.
If I may, when we get to it.
Okay. You want me to tell the grumpy
side
of the story?
Tell the grumpy side of ketamine, please.
Okay, grumpy side of ketamine. 25 years ago, I guess, whenever that was, 93 or so, my mentor, probably one of the smartest guys, and if not one of the most knowledgeable guys about neuroscience I've ever known, wrote a grant proposal proposing an NMDA theory of schizophrenia. It was beautifully written. This was like 20 pages, single space, and I read it. I was like, wow, this really could rock the world if people read this. And we were just proposing a series of studies that would just
putting in a proposal to the National Institutes of Health. And I would like to mention that these things are considered protected under strict confidentiality.
Oh, yeah.
Right? No one is to talk about them outside of the study section. You're certainly not to pick them up and run with them. You're there when you're on a study section to evaluate the scientific merit of the proposal.
Sure.
So when you submit a grant to the NIH, it goes to about a dozen people, maybe more.
It goes to a lot of people.
But we all sit around a table. I've been to a bunch of these.
I'll mention that I've sent in grants before that I send to one study section and I got the worst score, and I send the exact same grant with no changes to another group of individuals in NIH and got the best score. And this is out of a couple of hundred people.
I've done the same thing. You send in a grant, you send to one place and they're like, oh, this thing is awesome. They give you totally top marks, but we want to forward it to this other study section because they have more money, right? You get it to there. These guys are like, what, are you crazy? Did you fall on your head? This is terrible. And I think you told me that with Barry's proposal for ketamine,
For the ketamine,
they
for the
actually
NMDA
were
receptor stuff.
abusive, I think. In
They basically
response.
tarred and feathered him. I didn't know we were going to mention his name, but anyway,
I'm sorry.
They tarred and feathered him.
He's a real person. You can look him up.
It was a fictitious name anyway. So they tarred and basically, I mean, it was just a nasty, nasty review.
They kind of told you this is the lowest possible score we can give you, and if we could, it's like people saying,
we
I'd give
give
them
you a
1
worse
star,
score.
but if I could give 0 stars, I'd give 0 stars.
Or more
It's the
like
same
negative.
mistake.
Yeah,
negative
we
stars.
can give you a negative star.
So as well, not only did they opt not to fund it, which is easy enough to do, just one person in the study section has to.
Yeah, and this is a group of people that are big names in schizophrenia research.
They went out of their way to crush this idea.
Okay, so guess what starts percolating through the literature a year or two later?
Yep.
Year or two later, all of a sudden, the NMDA receptor hypothesis in schizophrenia starts percolating around from the esteemed people
who were sitting on this study section.
So this kind of crap happens a lot. And just let me say to these people, well, maybe I won't say anything.
No, that's
well, none of them would ever watch.
Yeah,
they all
right. Neither one of you, I'm sure, watching has been on an NIH study section. But I can tell you,
but this just gets me in a foul
thing.
mood when I hear about that. It's like, really, we couldn't even get funded to do this study.
I have had ideas stolen from proposals that I put into both the NIH and the NSF, and it's a really bad thing, and this whole system needs to be changed, because the current system of evaluating and awarding grants through the NIH and the NSF really gives the opportunity for very senior people who are sitting on these study sections to gain really early access to sensitive new ideas, and to then establish their priority with their new idea. And this is what has happened with ketamine, right?
Yeah.
So they basically, it came on NPR as if it was a new idea.
Well, it's percolating. Now, 25 years ago, it was just in the beginning stages of research. Idea gets percolated, gets put out there. A few researchers started doing some stuff with animal models, and then over time, it percolates out into the medical field, and some physicians started using the drug, just to treat different, for other, for different neurological disorders that the drug was not approved for. And then all of a sudden, a few years later, Johnson & Johnson starts developing their own nasal form of it, which, they're just doing this based on their knowledge of what's in the literature.
I mean, they're trying to do something.
But, you go to the very beginning, and actually, there probably was somebody 20 years before his proposal that probably made the same thing.
Somebody else probably did recognize this thing, and they put it out there, and it got squashed. And then the people that Mark was working with, they couldn't have known. They didn't hear about it.
Yeah.
And so the thing is, scientifically, there's this myth that, there's this one genius person sitting in a corner, and they come up with all these ideas. And while you do occasionally get, once or twice a century, you do get an Einstein. For the most part, the rest of us are all kind of like, sort of struggling to find something. And a lot of these ideas pop up. In fact, there's a whole website on this, inventions that were simultaneously patented in different countries. And so, it's virtually everything you can think of, because these ideas tend to sort of percolate up through a lot of different minds, and people are constantly talking and everything. But the problem with that is that when you put a single point regulatory control on what is acceptable and what is not acceptable scientifically, and then you use federal money to enforce that mechanism gets abused.
Yeah, and that's one reason that we're pretty enthusiastic about some crowdsourced science studies. And one of the things that we're going to talk about later is how we're going to organize that. Yeah. And the other thing we'll talk about are some N of 1 experiments. And basically what we want to do is have a large enough group of brain gauge users so that we can actually do some pretty solid scientific studies. And I can envision some combined brain gauge and pulse electromagnetic field studies.
Brain gauge with everything, nutrition and exercise and all kinds of different things. Because brain gauge can tell you, nootropics. We've talked about this a bunch of times. And if you're waiting for the scientific studies to come out of this, your grandchildren will see them. They won't necessarily be right, but they're going to be, a generation or two from now. And I think the solution to a lot of these problems is simply to put stuff out there.
Well, it's sort of like this. We could do some experiments where we actually show something that works.
Yeah.
And people could start, no matter what it is. People would actually have that information, and it would never get a federal dollar and never get anybody's stamp of approval
That's
or
right.
disapproval.
So we've gotten, I was going through some of our emails, and we've actually gotten comments where people are like, well, how do I run a study? And I need some help. Like, you know, of course, I don't have a footprint in academia or whatever. And the answer, I think, is to just start self-experimenting. If you know, you feel comfortable with that, you can actually start doing that sort of thing. And you can start doing N of 1 experiments. And it's, you know, some of these things you can kind of think yourself into a corner, and you can sort of overthink some of these. Sometimes you just simply have to try, you know, like, do I like broccoli? You know, I could I imagine I could structure a big study on that and everything, and eventually know the answer with a 95% probability in about 20 years. Or I could just try some broccoli, right? And that's basically, you know, I could, right? It's an N of 1 experiment.
You like it raw or cooked?
Well, I like it cooked myself. I like it really soft too. But that, you know, but I only know that because I've tried it, right? And it's sort of like, you know, some people are asking us, well, you know, some people are thinking a little bit too big, a little bit ahead of themselves. And I think grassroots science starts with an N of 1. You know, all of my research on PEMF and using it for orthopedic and chronic pain and stuff like that. That was, you know, the first many years of that was just me experimenting on myself to see what worked. So I think the answer is you just sort of, you start, and then of course, once you're doing something, Mark and I are happy to give you an opinion on it.
Yeah.
Right? And the thing is that, you know, there's no such thing as a perfect, clean, well, almost no such thing as a perfect one-off, perfectly clean, it's done perfectly the first time, and it's only done once experiment. You know, it's sort of like people have to kind of go around an idea like a shark, you know, going around a chunk of, you know, beef floating in the water. It's kind of like, yeah, kind of poke at it from a bunch of different directions. You try a few different experiments, you learn a few things. So these experiments are kind of like, you know, it's how the sausage is made, as they say in science, is that you have to try it a few times. A few times it's kind of, you know, doesn't work out well. You go, oh man, I wish I'd have known this or that. So you're way better off doing something small and informal, right?
Yep.
Giving it a try than trying to structure some big complex thing from the very beginning. And we think that, I think for sure, we think that crowdsourced science and grassroots science is going to grow up to be that way, where people just, you know, they're not worried about all the trappings that go with, you know, academia. They just simply want to know the answer.
Yeah. Did you want to tell your grumpy story, or you want to wait for the next video?
I'll tell my grumpy story really, really briefly. Okay, so about 20 years ago, I had a laboratory at MIT. It was 2000, 2001, so it's about 18 years ago, in the artificial intelligence lab. And one of the things I was working on was hybrid prosthetics, which is like, can you integrate tissues with synthetic things to make prosthetic limbs and stuff. And I had some funding from the Department of Defense. And one of the things I did as a demonstration was I actually built a robot. It was a robot that was powered by living muscle from frogs, and, you know, frogs actually that were being thrown away from a previous experiment. So I harvested these tissues, controlled them with infrared, and we were able to make them swim around and do all kinds of stuff. This is almost 20 years ago, right? So what's in the news this morning, the last 3 days?
A robot frog?
No, someone has finally integrated muscle. You know, it's like, you know, but these people, these scientists, they went way out of their way to like maximize their you know, their media exposure. And so far as I can tell, they made no reference to the work that I and others had done 20 years ago, which was much more sophisticated. They just had a muscle attached to a little thing, and it could move backwards and forwards. I mean, we built a robot you can control remotely with infrared that would swim around. You know, it was what we did was more sophisticated. It was almost 20 years ago, and of course they're acting as if nothing happened. Well, at the time, I was pretty conservative about who I would talk to in the media and everything. I got many media requests. You know, can we come and film in blah 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 from Germany, all over the world? And I was like, no, you know, I'm busy doing other things. But a lot of scientists are just kind of media hogs, and they just want to, you know, get in the news and get interviewed as much as they can, and they want to act as if they have a new idea. And I think that it is a safe bet that any time you see a scientist on the news, like on a network news, telling you that they've done something new, especially when it comes to tissue engineering or anything related to the brain,
ain't new.
It ain't new, and they didn't think of it. And that is, you could bet your house on that, and you know what you'd end up with? 2 houses. You know, you know, if you want to if you could find someone to take you up on that bet, that would be the bet to make, because especially in like tissue engineering and brain research and all these other sorts of things, you know, nutrition, all these things, most of the ideas that end up in the news, they have been around 20, 40, in some cases over 100 years. And it's just that the people who are presenting them to you now as their idea, they're not giving any credit to the people who came before. And that makes me grumpy.
Yeah.
I mean, I'm already grumpy. It just makes
some
me
of the
grumpier.
big things that are new never really get a big splash either. Like when guy discovered which cells in the brain are actually producing insulin.
Oh, yeah.
Just paper in a scientific journal, and that was it.
Because this guy's a real scientist, right? He's like, hey, this is an important discovery. I'm going to put it in a reasonable journal. It's appropriately scaled for the importance of the finding. Put it in there, and boom, that's what. So real scientists tend to do that. Kind of quiet. They do their thing. They sort of shun the media. Whenever you see a scientist who's like some sort of media rock star, they are full of it. I don't know a single one who's
Aren't we
actually
considered a media rock stars?
well, both of you, maybe all four of you who are watching
show.
us.
Yeah.
I don't know if it's two of you or four of you with like, alternate names. But, whenever you see a scientist who's like on the news all the time and you see them in their lab and they're moving red fluids between glassware and stuff like that, that person's not really a scientist. And if you really like just scratch the veneer, you'll find that they've got press release after press release. They may have hundreds of papers. If you take the sum total of all the work they've done, they will have spent tens of millions of your taxpayer dollars, but they don't contribute. And so this is kind of a case, this ketamine thing, but it happens all the time, practically every day, practically everything you hear in the news. It's not really news. It's not new, and you really can't attribute that new technology to the person who's taking credit for it. So that's a pretty grumpy 20 minutes. I think it's time to stop. Sorry about that. Well, no, I'm not sorry. You're watching Grumpy Science, right? I mean, you can stop at any time.
Yeah,
so if you're still here and you're like, why is that Bob guy so grumpy? You know what? It's on you because you didn't just move on to the next video like 10 minutes ago. All right. See you later.